We all know this paradigm of cancer evolution, evolving from a normal tissue to a premalignant tissue to one that's invasive to one that's finally metastatic. We know that if you look under the microscope histopathologically, there's characteristic changes that occur throughout each of these stages. And that if you look at the DNA, at least in this model of colon cancer, for example, you see accumulation of key mutations that either turn on or turn off genes in key locations that accumulate over this pathway of normal to premalignant to invasive. And we've pretty much defined these mutations that we measure in pre-cancer cells or cancer cells, but being absent from normal cells. So what do you do with all this information? Because last time I checked, I think there are over 3 million variants that are reproduced across a variety of different tumor types, and that comprises our somatic cancer genome data. And the real hope was that we could use these mutations and marry them to a therapeutic target, and the tumor would melt away. And that's happened, or there have been signals that that is possible with some therapeutics. And in some therapeutics, it works very well, and others not so well, but all dictated based on a genomic alteration and a drug that can specifically target that genomic alteration. But it hasn't been a tidal wave of therapeutics per se that have been highly effective. We've also used these markers as prognostic markers to tell us if a tumor is going to be more aggressive or less aggressive. BRAF in colon cancer is one such indicator. IDH in brain tumors is another indicator of aggressiveness or lack of aggressiveness of that tumor type. This is a world I know very well in liquid biopsies, and by here I'm defining mutations as a dynamic marker to look in bodily fluids for the presence or absence of cancer, for the growth of cancer, for looking for resistance mutations, or things like minimal residual disease. But I'm going to focus mostly on using these genomic alterations as immune antigens in a way to stimulate the immune system in a way that we can manage patients in a very positive way.